Hello. So today we talking about microscopy. So we'll be focusing mainly on the theoretical aspects of microscopy in today's lecture. So talking about a little bit of history. And Zacharias Hansen, along with his father Hans Hansen, is credited for the invention of the microscope around 1590s. But the use of microscope for exploring microbial life was given to Anton von Leeuwenhoek, which we all know, and it was around 100 years later than the invention of the microscope in 1670s. So there are different types of microscopes available which works on different principle. Each of them work on a different principle. So one is a bright field microscope, which we usually have in undergraduate labs. And there is dark field microscope, fluorescence microscope, polarizing microscope, and electron microscope. We'll be discussing about the principles of each one of them one by one. And after that, we'll be discussing about bright field microscope in detail. So coming to dark field microscope, what is like a dark field microscopy? In that there is a white light which is coming from the illumination source, but the light does not come through and through. There is an annular filter which is present in between, and that annular filter will filter out all the rays of light which are coming directly to the specimen. For example, all the light which is coming directly to the specimen will be filtered out and will not pass through, and only the peripheral rays of light will pass through. These peripheral rays of light will then go on to incident on the specimen, and then specimen will reflect the light into the objective. So whatever we see are the diffracted rays of light, and the non-diffracted rays of light will pass through and will not go into the objective, and the center light which was coming will also not pass through. So the background of the specimen will appear black or dark, and the specimen itself will appear like a brightly lit specimen on a dark background. This is commonly used for spirochetes. This is how dark field microscope works. Then fluorescence microscope. The principle of fluorescence microscope is based on the property of the specimen, the property of the fluorophore, which we added to the specimen, which would then bound to the specific parts of the specimen, which would be antigen or antibody reaction, whichever is happening. That fluorophore's property is that it will be excited by a particular wavelength of light, and after excitation, it would emit another wavelength of light, which would generally be longer than the excitation wavelength. There is an incident ray of light. Incident ray of light has all the wavelength of light photons. The excitation filter, what will it do? It will filter out all the wavelengths which we don't need, or which will allow only that wavelength of light to pass through, which will excite the fluorophore. There is a dichroic mirror which will reflect the excitation wavelength of the light. Then the excitation wavelength of the light will act on the specimen, and then the specimen will be excited and emit another wavelength of light. That another wavelength of light will be allowed to pass through by the dichroic mirror. That is the property of the dichroic mirror. It will reflect one wavelength of light, and it will let the other wavelength of light pass through. So the other wavelength of light will be allowed to pass through, which is the emission wavelength, and then it will be detected by the detector. Also, there is an emission filter which will filter out, or it will make the emission light more precise. Okay. So next there is a polarized microscope. Polarizing microscope, what is the principle of polarizing microscope would be? There is a light coming from the source, and then the polarizer will make the light plane polarized. So now the light is only in a single plane. This single plane polarized light will be incident on the birefringent specimen. That is the property of the specimen, that whenever a plane polarized light is incident on the specimen, it will make the light into two perpendicular lights, two perpendicular planes. So one would be an ordinary ray, and the other can be called an extraordinary ray, and then it will be analyzed by the analyzer. So that is the principle of polarizing microscope. Now comes the electron microscope. In electron microscope, instead of the lenses, which are the condenser, the objective lens, or the projective lens, whatever lenses are there, instead of the lenses, these are electromagnetic coils. So these electromagnetic coils will generate an electromagnetic field which will deflect the electrons coming from the source and make them incident on the specimen or the sample, and then it will also make the path so that the electron can go on and can be detected on the screen. So these are just the electromagnets which will create electromagnetic coils which will create electromagnetic field. That is how an electron microscope works. So now coming on to the compound microscope. Compound microscope works on the principle of bright field microscopy. What key? There is an incident ray of light which will be incident on the specimen, and then the specimen will reflect the light which will be detected by the lens system, and then we can see after the magnification. That is the basis of bright field microscope. So in a compound microscope, what are the broad parts in which the microscope can be divided are the supporting system. The supporting system is this, whatever is the base, pillar or the handle. Then there is a focusing system. The focusing system may or may not include the stage, but it includes the fine focus and the coarse focus knobs. Then there is an illumination system. The illumination system is from where the incident source of light will be coming. And then there is a magnification of the optical system, which includes the eyepiece and the objectives. We'll see one by one. So in the supporting system, there is a base, then there is a pillar is this, and then after the pillar, there is an arm, and then there is a body tube. The body tube connects the eyepiece to the nose piece on which the objectives are placed. Okay? This is the supporting system. Then there is the focusing system. In the focusing system, there is a stage. The stage, there is a mechanical stage and there is a fixed stage. The mechanical stage is one which can be moved, so that when we are focusing any specimen or a slide, we can move the stage to see different fields. That is the function of the mechanical stage, and we can place the slide there. And the fixed stage is on which the slide is placed. That is the fixed stage. Then there is a coarse adjustment and there is a fine adjustment screws for coarse and fine focus of the specimen. This is the focusing system. Then comes the illumination system. The illumination system, older microscopes consist of the source of light. Whatever is the source of light could be the natural source of light or LED bulb which is placed outside or an inbuilt on the microscope. That is the source of light. So then there is a condenser. The condenser is placed above the source of light and it condenses or focuses the light rays onto the specimen. That is the function of the condenser. The iris diaphragm controls whatever the amount of light is passing through it. The iris diaphragm will control the amount of light and condenser will focus the light on the specimen. So this was the basics of the supporting. Then there is a focusing system. Then there is the illumination system. But the most important thing in the microscope would be the magnification of the optical system. The optical system or the magnification system consists of an eyepiece. The eyepiece is through which we see, and there is also power of the eyepiece, usually 10x, so that will also be included in the magnification. Then there is a nose piece, which is usually a revolving nose piece, which we can revolve to change the objective which is into the focus. And there are objective lenses which are attached to the nose piece. So now going into details of the objective lenses. So these are the different objectives which are available routinely. So there is a 4x objective. 4x means that is the 4 times magnification or the power of the objective lens. 10x, then there is 20x, 40x, 60x or 100x. Most commonly, the objectives which you will be seeing would be 4x, 10x, 40x and 100x. 10x is usually called the low power magnification. 40x is the high power magnification and 100x is the oil immersion. That is, you can see that it is written here oil. So this is an oil immersion. We'll talk about in detail. 4x can also be used. It is called as the scanning objective. So 4x is used for scanning the field, broadly scanning the field, whatever is there. And then the low power magnification, we can see the uniformity of the structures and which area to focus. Then high power microscopy, we can see the counting of the cells, and especially in the Newbar's chamber, we can count the cells. And in 100x microscopy or the oil immersion, we can see the morphology of the cells in detail. So this is what the objectives are. And now we can see what is written on all these. So 10x would be the power. Then there is 0.25. 0.25 is the numerical aperture, which we'll be talking about in details, but you can see that the numerical aperture is increasing as the power is increasing. That you should remember. Then there is infinity, which is written, that is the infinity corrected lenses, that there is another lens which is present in the body tube which will correct for infinity. That means that the light rays which are coming from the specimen will be reaching the eyepiece as if they are coming from an infinity. That is what infinity correction is. And then there is 0.17, which is written in some of them, which means the cover slip thickness which should be used. So now, in newer microscopes, there is a system known as parafocal distance. So the parafocal distance, what that is, in parafocal distance, for example, this green light, this is the tube length of a 4x objective lens, and this is the working distance. The working distance is the distance between the objective and the specimen which is placed. So this is the working distance. But this is the parafocal. The distance between the nose piece and the specimen is the parafocal distance. Now seeing the 100x microscope. The 100x lens, the tube length is increased, but the working distance is decreased. Tube length is increased, but the working distance is decreased. That is what is known as the parafocal system. That the increased tube length is proportional to decreasing working distance, so that the distance between the nose piece and the specimen always remain the same. So what is the benefit of that? The advantage of that is that if you focus a specimen under 10x, you don't have to focus it again on 100x. The working distance will automatically change according to the lenses. Okay, so once you focus on 10x, then you don't have to focus again on 40x or 100x. You just have to change the fine focus. Fine focus has to change, but the coarse focus does not have to change. That is what parafocal microscopy is. Okay, and also the working distance can sometimes be written on the objective lens themselves. The working distance will decrease. The numerical aperture increases with increasing power, but the working distance decreases with increasing power. So now coming to the how the image is formed. So the rules of image formation. Now, first of all, why is it called a compound microscope? It is called compound microscope because there are two lenses. There are two lenses which will be forming an image. So one lens is the eyepiece and the other is the objective lens. So that is why it is called a compound microscope. Now each lens will form an image according to their properties. The property of all these lenses are convex lenses only. So property rules of image formation would be an incident light ray which passes through the optical center, does not suffer any refraction. We'll be seeing how this happens. An incident light ray which is parallel to the optical axis is refracted passing through the focus, and which the incident light ray which passes through the focus is refracted parallel to the optical axis. So what happens, for example, in a convergent lens? In a converging lens, if we see how the image is formed, for example, this is the focus of the lens and the object is placed behind the focus. So now the incident ray of light which is parallel to the optical axis will pass through the focus. The incident ray which was passing through the focus will become parallel, and which was passing through the optical axis will go as is without any refraction. They'll intersect here, and then the image of the object which is real, inverted and magnified will be formed. This is how an image is formed in a converging lens if the object is placed greater than, the distance of the object is greater than the focal length. Okay. Now what if we place the object within the focal length? So we place the object within the focal length, the line, the incident ray which was parallel to the optical axis, so the incident ray which was parallel to the optical axis will pass through the focus. The incident ray which was passing through the optical axis, optical center will go as is, but they are diverging rays of light, so they won't meet. We will have to plot them behind the lens, the imaginary lines, and they will join together behind the lens. Still the image is magnified, but it is virtual and erect now because they are not joining at a real point anywhere beyond the lens. Now in a diverging lens, what happens is, that was a converging lens. In diverging lens, what happens is the incident ray of light, which was parallel, for example, this is the object. So the incident ray of light which was parallel will pass through the focus, so it was diverging, and the incident ray of light which was going to pass through the focus will go parallel, and the optical center will go as is, and will form a object which is smaller than the original, the image which is smaller than the original object. Okay, so this is how image is formed in diverging lens. We don't need this in the microscopy, but just for completion's sake I have told you. So what happens is, the image of an object in a converging lens, as we saw, if the distance of the object was less than the focal length, what was the for image formed? The image was virtual, erect and magnified. If it was between focal length and two focal length, matlab f and 2f, then it was real and magnified, which we saw, real inverted and magnified. But if the object is placed at 2f, then the image will have the same size, and if it is beyond 2f, then it will be demagnified. But in diverging lens, what happens is anywhere you place the object, it will always be demagnified. Okay. So in microscopy, what we'll be needing is the first two which we discussed. Either the object would be placed within the focal length or 2f, or the object would be placed within the focal length and the image would always be magnified. Okay, so that is how the magnification increases and is multiplied. If the image is magnified by the objective 100x and it is then again magnified by the eyepiece 10x, so the total magnification comes out to be 1000x. So how is the image formation happening in a compound microscope? So for example, this is the objective lens and this is the eyepiece. The objective lens has more power, so the focal length is less. The eyepiece has a lesser power, so the focal length would be more. This is the focal length of the objective, FO and FO dash, and FE dash would be the focal length of the eyepiece. Okay. Now for taking into account the object is placed, the D object, distance of the object is within the 2F and F of the object. Object is placed more than the focal length but within 2F of the objective. So the same thing happens. The image which is formed is real, inverted and magnified. Now this real inverted magnified image of the object is now within the focal length of the eyepiece. Now it is within the focal length of the eyepiece. Now the eyepiece will make a virtual and erect and magnified image of this image which was formed by the objective, which is this. So this is the final image which is formed, and this image we can see through the eyepiece. This is where our eyes are, and then we can see this virtual and inverted image which is magnified twice of the original. So this is the ray diagram of the compound microscope which you should remember. Now coming into the numerical aperture which was written on the objective lenses. So numerical aperture was keep increasing with power. The more the power of the lens, more was the numerical aperture. What is actually the numerical aperture? It is the capability of the lens to gather light. Light gathering capacity of the lens is numerical aperture. So how do we calculate that? Numerical aperture is n sine theta. N is the refractive index between the medium between the objective and the specimen, and sine theta. Theta is the half of the angle when a cone of light is made by the object by any lens. So if a cone of light is made, maximum light cone, and half of that angle would be theta. So that is the numerical aperture. So now taking the example of 10x and 100x microscope. 10x microscope has, well, objective, sorry. 10x objective has lower magnification. So the focal length would be more. So if the focal length is more, the angle which would be made, what would be the angle made? Angle made would be less. So n sine theta would be less. So the light gathering capacity is less. As the power increases, the focal length keeps on decreasing, but the angle keeps on increasing. So n sine theta also increases, so numerical aperture increases. So that was about the numerical aperture. Now there is a concept of limit of resolution. Why are we actually doing the microscopy? Why we are doing it? So as to magnify first the specimen which we need to see. And also along with magnification, what is more important is the limit of resolution. Resolution refers to the smallest distance between two distinct objects which they can still be distinguished at separate points. That is similar to like 2-point discrimination. Like if I put 2 points close to each other, what is the minimum distance in which you can tell that they are different points? That is what a limit of resolution or resolution is. This is in centimeters, micrometers or millimeters. Now limit of resolution would be 0.61 lambda by numerical aperture with respect to microscopy if we are considering. So as we can see that the limit of resolution is very much dependent upon the incident ray of light and the numerical aperture. So as we can see that limit of resolution of our eye is 0.2 millimeter. That if I put 2 points within 0.2 millimeters of the distance, my eyes can discriminate both of them as two separate points. With light microscopy, that limit of resolution increases to 0.2 micrometer. But I cannot increase it to much more even if I have a higher power magnification. Even if I take 150 or a 200 power magnification, 0.2 micrometer would go down to 0.15 or 0.13, because then the limiting factor comes out to be the lambda, which is the wavelength of the incident light, which will always be within the visible ray, which is 400 to 800 nanometers. But in electron microscopy, we are using incident light as the electrons, and then the lambda decreases a lot and the limit of resolution can go down to as low as 0.5, practically 0.5 nanometers, but theoretically it can go down to as low as 0.1 nanometer also. So even if I take a 100x magnification, the light microscope can resolve only 0.2 micrometer because of the wavelength of the incident light. Okay. Now we have discussed some of these things like power. Power is inversely proportional to the focal length. More the power of the objective lens, less is the focal length. Now power is inversely proportional to focal length. Then we saw that the lesser the focal length, the more would be the theta and the more would be the numerical aperture. So more the power, more the numerical aperture, and power is also proportional to resolving power. Resolving power is the ability of a lens to resolve two objects which are close to each other. But resolution is inversely proportional because resolution is in micrometers, which we talked about. The minimum distance. So if objective has a more resolving power, that means it can resolve two objects which are closer to each other. So the resolution would be less. You understand this? Okay. So 100x microscope will have more power and less focal length, will have a more numerical aperture, more resolving power, but the limit of resolution, which is the minimum distance between two objects which can be separated, would be less. Okay, understood? So now coming to some of the practical aspects of how actually you are going to focus the specimen under microscope. So there are three things which you have to take into account. The first important thing is you have to always focus under low power first and then go into the higher power. First thing to take into account. Second is that this is a parfocal system. So whenever you are going from low power to high power, you don't have to change the coarse focus. You just have to change the coarse focus, or when you are focusing under low power, after that you don't have to change the coarse focus. And the third thing is the position of the iris diaphragm and the condenser, which is the most important. Sometimes you can be focusing the under 40x or 10x very correctly, but the position of the condenser or the iris diaphragm is not correct. So the light coming is not correct. Either it will be very brightly illuminated or it would be very dimly illuminated. So that will not be, you will not be able to see the specimen under focus. So the steps would be: first, you should place the specimen onto the fixed stage and the mechanical stage. So you should put the specimen on the stage. Then place the stage to the topmost position, seeing the objective lens. See, you can do it either ways. Either you can go from up to down while coarse focusing, or you can go from down to up. The benefit of going from up to down is that you are never at the risk of damaging the specimen or the lens, even if you go too close, because you are going down. So first you will be in the uppermost position. The stage would be uppermost, but the iris diaphragm would be partly open, just open, and the condenser would be lowermost. Then you will focus under the lower power microscope. Then you'll be switching the lens to higher power, which would be the 40x, and then you can just by fine focusing and changing the iris diaphragm and the condenser, you can focus it under the 40x. After seeing the area under 40x or counting, then if you want to see a morphology or better magnification, then what you'll do is you'll switch the nose piece and put it into middle so that no objective is in focus now. Then you will place a drop of cedarwood oil onto the specimen, and then you will click the 100x microscopy. The iris diaphragm will be fully open, the condenser would be uppermost position, and then you'll just use the fine focus to focus under the oil immersion. Now, why are we saying it oil immersion? Because we are using cedarwood oil. Now this is understood, but why are we using cedarwood oil? Cedarwood oil is used because the refractive index of the oil is similar to that of the material of the lens. So Whenever a light ray passes from a denser medium to a rarer medium, the light rays would diffract. So if the light rays were passing from the specimen to the air in between the specimen and the objective, the light rays would diffract and all the rays of light would not go into the objective. So we place a drop of cedarwood oil so that there is a single medium between the specimen, the oil and the lens. So now there is no refraction of light and all the light which was focused by the condenser onto the specimen, because it was in the uppermost position, it was condensed onto the specimen, will directly go into the objective, which has a very high numerical aperture. Numerical aperture high means that it has a very high light gathering capacity. So all the light which is coming onto the specimen focused by the condenser will go into the objective and then will be able to see the object more clearly or the specimen much better. Now what is the use of iris diaphragm? Fully open iris diaphragm will allow more light to pass through and so that the specimen can be seen very well. So that is how the basics you focus the microscope, and after using the microscope, especially the oil immersion, you should definitely clean the lens, because it is much easier to clean the lens when the oil is wet than the oil is dry. After that it is very difficult, and you should handle the microscope very carefully on the pillar and the second hand below the base. That is how you should handle the microscope if you are keeping it somewhere else. And the use of the binocular eyepieces is so that there is lesser strain, and these binoculars can be changed so that the interpupillary distance can be adjusted according to your needs. So this was all about basics of microscopy. If you still have any doubts, you can ask them in the comments or in any of these social media handles, and we'll try to get back to you as soon as possible. And we'll keep posting more videos. Thank you.